This is a production of Cornell University.
Okay, great. Yes, no, thanks for the invitation to do this. It was very nice to meet way back earlier in the year, Libby, over in Cambridge. So yeah, the presentation I'm going to give you today is entitled Defining the Core Infection Biology of the Wheat Pathogen, Zymoseptoria tritici, through multi-omics approaches. So I'm a member of Rothamsted Research that I'll introduce in a moment in the Department of Protecting Crops and the Environment. There we go. Okay, yeah. So in answer to the question about how old we are, um, as far as I'm aware, we are one of the world's oldest agricultural research institutes. So we are based about 20 kilometers north of London in the United Kingdom. We were established in about 1843 by the gentleman in the top left corner of the picture there, Sir John Bennet Laws. And as such, we've been sort of hosting some of the longest continuous agronomic experiments that are currently known. And a notable one is one we call on a field called Broadbalk, which is a winter wheat field. And that experiment has been pretty much running as long as Rothamsted's been going, since about 1844. And the ladies you can see on the picture there have been storing away wheat samples from that particular experiment, so vegetative tissues, root tissues, soil, etc., year on year from that date for us to analyze at any point in the current time and still going forward. But I really just show this slide to highlight the fact that wheat has been a major focus of Rothamsted's research for a long, long time, and it continues to be so. So in the presentation I'm going to give today, I'm going to start with some key aspects of the pathogen and the disease problem, introducing it. And then I'm going to give you 5, if time permits, mini presentations, if you like, mini scientific research areas that we're working on, starting with looking at pangenomics as a method to identify core genes for functional analysis. I'll give an update on some work we've been doing on characterization of a core virulence infector protein from the pathogen. I'll introduce what I call the fast-forward genetic screen for virulence gene discovery, with a focus currently on cell wall. And then I'll touch on what we call the Abraham-Wald essential gene screen and the associated mutant collection. And at the end, I'll shift tracks a little bit and talk about some work that we've done using this fungus as a tool for the discovery of new microbe-derived crop protection products. Okay, so the most important foliar wheat pathogen of wheat in the UK is Zymoseptoria tritici. It causes Septoria tritici blotch disease, which you can see in the picture in the middle there. You end up with necrotic lesions bearing asexual sporulation structures waiting for rain splash to propagate the disease onto new areas of the vegetative tissue. So the ultimate cost of the disease is through loss of photosynthetic capacity. And this is currently the #1 disease problem for UK and European wheat farmers. To put that in perspective, current calculations have suggested that 70% of all fungicides that are applied on arable land in Europe, in Western Europe in particular, are applied principally to control this disease. The farmers really do not want this disease to get into their wheat crop. So that is his economic and environmental footprint. It's a representative species of the globally important Dithidiomycete class of plant pathogens. And importantly, really, I think for today's presentation and for the system as a whole, is it's long been regarded, long been known to be a rapidly evolving pathogen with a highly flexible sexual reproductive system, very unstable genome, lots and lots of ways of shuffling the deck in terms of genomics. And it's been a model system for looking at pathogen population dynamics well before we ever sequenced any pathogen genomes. So it's a model organism for rapidly evolving species. And it's haploid in all but its sexual phase, which aids some of the work which you'll hear about today that we do on it. And in terms of controlling this pathogen, obviously due to this rapid evolution, there is poor host resistance, and there is whenever new fungicides are applied, resistance occurs very, very quickly and gets fixed and is prevalent in populations. And there currently is not one single fungicide on the market, if used in isolation, is effective against this pathogen. So basically, what we need are new disease control targets, and our research focuses on how we can identify the weaknesses that could form those targets. So to just give you our overall research objectives and our methods, basically we need to understand, or we're trying to understand the genes and pathways which enable leaf infection. But also, more recently, we've been starting to look at the potential genes and pathways that might be essential for life, because these are also potentially useful targets for disease control. So therefore, we focus on things that we think will hopefully prove useful in the mid, near, or long term for future disease control interventions, whatever they may be. The sort of methods we use are comparative genomics and bioinformatics. We use functional genomics and transformation, so we can make knockouts, we can overexpress genes, and we can very quickly and easily generate random mutant libraries, and I'll show you some of that data later. Whole genome sequencing is now very affordable, so we can use that to identify mutations and to look at the variation in pathogen populations between strains, but for pangenomics. And then we obviously conduct infection assays, do microscopy, conduct biochemical characterization, and so on. And one of the key things that we've been exploiting in our research is a very unusual characteristic of this particular fungus, in that it's classified as being dimorphic. So in certain growth conditions, this particular fungus will grow almost as yeast-like colonies, which you can see in the left-hand side of the slide there. And these are not directly plant pathogenic, because to become a pathogen to infect wheat leaves, these cells must germinate hyphae to go in through stomata to start colonization. And that can be triggered also in vitro by switching those colonies onto low nutrients or high temperatures on low nutrient agar, for example, water agar there, which simulates effectively what happens on the leaf surface. And those yeast-like spores start to germinate hyphae, which enable the fungus to penetrate the stomata and begin the early phases of infection. So this is a very simple laboratory assay, which forms a very good basis for a genetic screen to identify core virulence genes. Basically, if your particular mutant of this particular fungus gets locked in the yeast-like state and cannot transition into the hyphal growth state, you've pretty got a good chance that it's not going to be pathogenic on wheat. So you're getting into virulence genes. Just to reinforce that and make it very, very obvious and clear, is that what this also enables gives you great power for looking at genetics and genomics, whereby if you look at a single standard agar plate for Zymoseptoria tritici, you can have hundreds to 1,000 genotypes, whether they're natural populations or induced mutants. Whereas a similar plate for most of the fungi that you might be aware of, you'll have a single genotype colonizing an entire plate. So it really does bulk up the power you have for doing functional genomics types analysis on this system. The other important feature was that obviously we've had fully sequenced genomes for some time. We tend to work on this one, which is one of the globally accepted reference strains of this pathogen, and it's called IPO323. You can see here the fully finished genome of IPO323, which was done, wow, some time ago now by the JGI over in the US. And it was, at the time, it was the first complete finished genome for any fungus. So quite a scoop, really, for what was then called Mycosphaerella graminicola, before it now became more recently Zymoseptoria tritici. You can see there all the in the red bars, all the different genes that are predicted and where they are on those chromosomes. And you can see for this particular isert, we have 21 chromosomes. Now, an interesting feature and slightly confusing feature of this organism is that those small chromosomes, 14 to 21, can be lost and gained in natural populations. And they can also be lost in experiments, as I frequently find, which drives me a little bit crazy. You can start an experiment with IPO323 with 21 chromosomes, and you can finish your experiment even though you're not trying to affect the chromosomes with 20 or less. So these are highly unstable, and therefore they're likely to be dispensable for virulence, because natural wheat-infecting strains of this fungus can lack any number of those small chromosomes. Another important feature, which is a bugbear of mine and what I really want to get into going forward, is that typical of most filamentous fungi, actually typical of most eukaryotes, if you like, is that still 40% of the coding genes present in genomes are of unknown function. So these are the sort of things that are predicted hypothetical proteins, proteins with a domain of unknown functions, etc., etc. So there's a lot of stuff we don't understand about fungal genomes and probably eukaryotic genomes in general. And this is something that I'm trying to address in future, and I'll hopefully show you how we plan to do that going forward throughout the presentation. So the other key aspect for the work we do is to look at the virulence assays of the fungus on wheat leaves. This was one of the better experiments we ever did way back in the day, where we set up an attached leaf infection assay, which you can see here is where we put spores on the surface of a wheat leaf, and we can photograph it every single day. In this case, it's from day 14 to day 18. And what you can see is this very long period of symptomless colonization at the start, where you're not actually sure whether you put the fungus on, you think it's not going to happen at all. But then typically around about 10, 11, 12, 13 days, you start to see some discoloration and collapse of the tissue. And by 14 day onwards, you've seen that very same area where you applied the spores on completely collapse and go necrotic and dry and bear those pycnidia, those fungal sporulation structures. So the way we set this up, obviously, it allows us to do many different quantitative measurements for aggressiveness of strains, for host resistance, but also to test induced mutants of Zymoseptoria that we make in the lab for any loss of virulence or reduction in virulence. And we can score that by any change in the time taken to disease symptoms, or any difference in the leaf area where we inoculate that actually establish symptoms. And also we can measure the levels of fungal sporulation in the tissue simply by washing spores from the tissue or by qPCR methods. So a very quantitative assay that again forms the basis of what we do, much of what we do. Okay, now I'm going to touch on one of the first mini scientific areas that we've been working on, and this is unpublished data. Actually, no, it is published data, this one. Some of the other stuff is unpublished. So this is exploiting pathogen pangenomics to prioritize core genes for functional analysis. So whether or not you're aware of pangenomes, but everybody now is starting to generate pangenomes for whatever species they are working on scientifically, biologically. And a pangenome, in summary, is the sum of all genes that you might expect to see in the entire species, basically. So each individual will have a core set of genes, but then each individual will contribute its own unique set of genes to the mix. And if you sequence enough individuals, you will start to reduce the size of the core, if you like, set of genes that are present and functional in the entire species. But you will also start to uncover some of the genetic variation, which we term accessory genes, that potentially allow the selective interactions of pathogens, for example, with changing environments. And the larger the size of that accessory or unique fraction is, the greater adaptility of the species is thought to have to change to environmental stresses. But I thought about this in a different term. So what I really wanted to do, and you realize that Zymoseptoria is going to be super diverse, and it's going to try and shuffle the deck as often as it can. But surely there will be stuff in there that it can't shuffle. So the idea for us was to try and define and to harden down on what is the core genes. So the likely gene set that are indispensable for the lifestyle of all the individuals of this pathogen, because we thought within there could be some interesting new potential targets for future disease control. So we did a little pangenome analysis, not as big as some others have done, but a very focused and targeted one, which we started with reannotating the IPO323 genome, which needed a little bit of work. And then we took genome sequences of 17 other European isolates that we collected as far west as Ireland and as far east as Poland. We pathotyped them all on wheat genotypes and selected very aggressive strains. And then we also did RNA sequencing on those strains, or the majority of those strains as well, including in culture and during infection. And effectively, we built a pangenome, which was about just short of 18,000 genes from all this data. And then we assigned them as either core or accessory. So the core genes we assigned as being so due to being present in all isolates with no loss of function mutations in any of the isolates, and they were all expressed by all of the isolates. So we didn't have any particular strains where a gene was present but had no expression support. On the contrary, something was deemed accessory if it was missing from one or more isolates, had a loss of function mutation in one or more isolates, or had no expression in one or more isolates. And the key point about this is that you can see the size of the accessory chromosome is almost as large as the core, and this fits well with other pangenome studies done on this pathogen, and again reinforces the fact that this is a species that is capable of rapid evolution. It's a very large accessory gene set. But we have started to then drill into the core genes, which was our initial objective. So then what we started to do was start to do something which was perhaps a little bit crazy and simple at the time, but it's kind of worked. And to start to look at variation, chromosome by chromosome in terms of gene variation expressed as chromosome by chromosome, and gene variation expressed as genes, to compare and contrast, firstly, core and accessible chromosomes, but then also all genes. So we did this by looking at the mutations we had in each newly sequenced genome versus the IPO323 template, and did a simple SNP-F mutation calling, which gives you 3 outputs for every gene and every mutation, in whether it confers a low effect, i.e. the DNA sequence changes, but the amino acid stays the same, whether there is a moderate effect on the protein, DNA sequence changes, which changes the amino acid, or whether there is a high effect. The DNA sequence change results in a loss of function mutation, such as a premature stop codon, a frame shift, etc. And we just thought we'd have a look what this looked like. So looking at it on average chromosome level data was very, very clear in a way. When you looked at all combined different types of mutations, high, moderate and low per gene, averaged over the chromosomes, which is the top graph there, you can see, okay, there was a little bit more variation in the accessory chromosomes of the core, but not very much. But once you removed the low effect mutations, which don't induce amino acid changes, you started to see the blue bars in the second graph down be much higher in the accessory chromosomes than the core chromosomes. And then when you remove the moderate effect mutations and go down to the orange bars, you can see that pretty much the percentage of genes with high effect mutations on those accessory chromosomes are way, way higher than the average number present on the core chromosomes, 1 to 13. And you get a mirror image when you look at the gene expression of every gene averaged out on those chromosomes, whereby those small chromosomes hardly express at all relative to the average expression on genes on the core chromosomes. So this was a clear compartmentalization, which we'd suspected and had been partially reported before, but a clear compartmentalization of the function and structure of the core and accessory chromosomes of the pathogen. We then started to look at the individual genes, thinking whether we can actually start to select genes for analysis based on the fact that they might have lower mutations than expected, meaning the fungus is not really shuffling the deck in their particular case. And just to test this out initially, we started doing some data mining on all the published genes that were available for Zymoseptoria, including our own. And we divided them into 3 categories, and we looked at all the genes that were experimentally shown and published not to be required for virulence, those that have been required for virulence, and also we defined by orthology the predicted essential for life genes that are known from Aspergillus fumigatus and drew them across to Zymoseptoria tritici. And then we looked at the protein level polymorphism in terms of moderate SNPs that we can find in each of those. And we found a very simple result here, is that those genes that are not required for virulence had higher levels of polymorphism in the population than those that were required, or those that were predicted to be essential. So this gave us a sort of idea, is that can we use natural protein sequence polymorphism to prioritize genes for functional analysis? Can we actually identify some important new genes by looking for low levels of polymorphism? And to test this in the lab, we had a tDNA fungal mutant. So we generated this by random tDNA mutagenesis some years ago. It was called 2310. So it was the 10th colony picked from experiment 23. That's the basis of the name. And way back, we knew that this particular mutant, although it could grow perfectly well on rich agar, and it could generate hyphae, it could not generate hyphae on poor nutrient agar, and it was severely impaired in virulence on wheat relative to the wild type, as you can see on the left side of the slide. So we did a whole genome resequence of this particular strain, and we identified a 13 kilobase tDNA associated deletion, which effectively removed 5 genes from the genome of this pathogen. Those genes are shown there. So potential CYP51, cytochrome P450, a methyltransferase, transcription factor, a nucleoside diphosphate kinase, and a glycosyl hydrolase is all gone in the 2310 mutant. And then when we started to look at the pan-genome mutation rates amongst those 5 genes in our mini population, we started to see quite clearly that gene 4, which is the NDK, had far less sequence polymorphism between strains than the other genes present in that cluster. And conversely, it was the most expressed gene on average of all the genes in that cluster. Okay, so that was interesting. So we went, does this mean anything in terms of if we start to reintroduce those genes one by one into the 2310 mutant? Can we actually restore virulence by reintroducing any one of them? So each gene was reintroduced with its own promoter back into the 2310 mutant by transformation. And very nicely for us, I suppose, is that reintroduction of gene 4, the NDK, the nucleoside diphosphate kinase, was sufficient to restore full virulence to the 2310 mutant and also restore the hyphal growth defect seen on water agar. So that was kind of nice, and it supports our idea that we're currently running with, that we can use pangenomics as a useful predictive tool for gene functional prioritization. And one of the elements that we're currently using this in, that I'll come back to later, is to see whether we can really get into core genes with current unknown function that have low levels of variation. Okay, moving on to a second little mini topic, which follows on from the pangenome analysis. Tell you a little bit about the identification and characterization of a core secreted virulence effector. This project started some years ago, but we've updated it a little bit recently, and I think it's potentially interesting. So there was one secreted, a gene encoding a secreted protein with the lysem domains that is consistently and very reproducibly strongly expressed during infection by all strains of Zymoseptoria tritici. What you can see on the right side of the screen there is what I call a wagon wheel plot of relative gene expression. On the very outer rim, the numbers represent the different strains we've tested. The pluses say that it was present and functional in all of those strains. And then the C6 and 9 are the expression data for growing in culture, growing at 6 days during plant infection and 9 days of plant infection. So this particular lysem secreted protein was very, very consistently and nicely expressed in the same way by all strains and is a core gene. So we called this some years ago ZT3 lysem because when you actually look at the protein structure, it has a signal peptide for secretion, and then it has 3 lysem domains, which are potential carbohydrate binding domains, and those are the only real features of the protein. Many years ago, in collaboration with Bart Thoma, we characterized that protein and showed that it is actually a chitin binding protein. So chitin being one of the major constituents of the fungal cell wall, which is also a PAMP in terms of plant defense response induction. And ZT3 lysem binds very specifically chitin and not other complex carbohydrates. And we also showed some time ago that if you introduce it into a chitin elicitation assay, the protein, that it will dampen down that elicitation. So it is a well-known chitin binding protein. When you knock it out in the fungus, we also saw really quite significant reduction in virulence, which you can see in terms of the infection process, in terms of green tissue. that is left at 21 days post-infection, and also the fungal biomass that accumulates in that infected tissue. So we've long known it was quite a major virulence factor for wheat. We did some other work on this, which I'm not showing today, but basically we showed that if you were to silence the putative wheat chitin receptors, which are called SERC1 and CBIP, then the ZT3 lysem mutant could infect again. So it was a clear gene-for-gene relationship between one single effector present in the pathogen and some cognate receptors for chitin that were present in the plant. And what this also told us that basically the success or failure to induce disease is pretty much a straight battle between wheat and fungal chitin binding proteins in this system, which again is something that is potentially tractable in terms of disease control. What we've since gone on to do is ask a little bit more fundamental questions in terms of lysem effector proteins. So this was work done by, I think you would say graduate student, or I would say PhD student. She did some work looking at the orthologs of lysem effectors throughout the fungal kingdom. So these are basically lysem domain containing proteins with signal peptides for secretion. So they're likely to be secreted out of fungal cells for whatever reason. But you can actually find them in fungi with various different lifestyles, including saprophytes, endophytes, obviously pathogens. You can also find them in both the basidiomycetes and ascomycetes, and even in some of the early lineages of fungi. In different types of numbers, basically, depending on the species. But one of the key questions, there's been many reports on the importance of lysem effectors in plant pathogen interactions and even plant endophytic interactions. But one question we had is whether or not they could be functionally interchangeable between species, particularly if they kind of structurally look the same. So I let her go and pick some lysem effectors from her analysis that we would do some functional work on, as long as she also picked the Cladosporium fulvum ECP6 protein, which was the first lysem effector identified in any plant pathogen from Cladosporium fulvum by Bart Tomer and colleagues some years ago. And that we had to include in the project because it's very structurally similar to the ZT3 lysem if you do AlphaFold predictions, etc., which you can see boxed up there. But then I also let her pick out some other lysem effectors from with different structures and from different species, including some from G. graminis, which is Guanomyces graminis, which is a root-infecting wheat pathogen. So she picked a couple from there and a couple of other species from literature, a couple of other candidate effectors from other species. And then we basically decided to try and test whether there's any evidence that putative lysem effectors could be functionally interchangeable between species. So we did this by effectively taking all of those candidate effectors, hooking them up behind the 3-lysem promoter with a 3-lysem terminator behind them, and transforming them back into the ZT3 lysem mutant, and then testing whether or not they regained any virulence on wheat. And in a nutshell, this is where we are with this work, is that only the 3-lysem protein, when reintroduced into Zymoseptoria tritici lysem mutant, can restore full virulence. Intriguingly, ECP6 from Cladosporium fulvum, despite looking structurally very similar, cannot do that, and neither can any of the other tested lysem effectors thus far. So we have a little bit more work to do on this, but it starts to suggest that there might be specialization in lysem effectors in plant pathogen interactions, perhaps to deal with the different types of chitin structures that are made during those interactions, or the way chitin is bound during those interactions. So it's not so easy as just moving them between the two. They've clearly evolved specialization. Okay, now we're going to move on to the next little mini topic, which is a fast-forward genetic screen that we've been running for virulence gene discovery, and I'll try and tell you why I think it's fast-forwards initially. So one of the very simple and easy ways we can generate large collections of mutants in Zymoseptoria tritici is to zap them by UV light in a transilluminator, if you like. And this generates a lot of phenotypic diversity quickly and cheaply without any antibiotics, etc. And you can just see two examples here of the sort of diversity you can really get just from UV light exposing IPO323. On the left there are lots of pigmentation mutants where you have dark, pink, white albino mutants coming through very simply. And on the right, you can see the sort of variation you can get in hyphal growth when you start spotting these different mutants out on water agar. So this is really simple, really cheap. It allows you to build up huge mutant collections very quickly, and again is a good basis for establishing forward genetic screens. So the fast forward genetic screen that we've adapted thus far uses this technology basically, where we UV mutagenize our standard sequenced strain, and then we select for whatever particular phenotype of interest it may be, either strains that cannot transition into hyphae from yeast-like cells or directly on plants, although that takes a little bit longer, as you might expect. But whenever we get a mutant of interest, we do whole genome resequencing and SNP-indel analysis, because it's very cost effective, about 80 UK pounds per strain to do this at the moment, to see what genes we have mutated, whether there's any interesting genes in there that come out of multiple strains that become strong candidates, and then we start to investigate them in the lab via an independent knockout or a complementation and repeat the phenotypic check. And the first way we adapted the screen was to see if we could form a hypothesis around whether fungal cell wall mutants would be affected in virulence on plants. The reason we did this was basically because when you look around the literature for plant infecting fungi, there are remarkably few model systems anyway, actually, for fungal cell wall study, outside of looking at classic yeast, Saccharomyces cerevisiae. The three major species are Candida, Cryptococcus, and Aspergillus fumigatus, which are all opportunistic animal pathogens. They are either ascomycetes or basidiomycetes. So basically, there isn't still, I think, an intensively studied cell wall model for any plant-infecting fungi, and I thought it would be kind of nice if we could try to establish Zymoseptoria tritici in this area. And as a first step towards this, we obviously needed to generate a collection of mutants in cell wall strength and viability to see what's going on. So we did this using literally a very simple assay that was developed many, many years ago for yeasts, which is to test sensitivity of induced mutant strains to the stain calcofluor white. So calcofluor white has been used in yeast and in fungi as a screening tool to identify cell wall mutants on the basis of that if a particular mutant strain has a defect in its cell wall strength, it cannot grow back on calcofluor white despite being able to grow back on media. It can without calcofluor white, basically. So it's a powerful screen, well known in literature, and I thought we should be able to adapt it to Zymoseptoria tritici. So this is what we did, basically. This is a classical yeast replica plating screen, which we've used for the yeast cells of Zymoseptoria tritici. So we've done a UV treatment of the single cells to a 95% kill rate. We let them grow up a little bit on a plate, and then we print them onto black velvet, if you like, which makes a little imprint, a marker of what was growing on the plate. And then onto that black velvet plate, you print your control plate and your test plate, which has calcofluor white in it in this particular case, and you grow them up. And then effectively you look for colonies that grow back on the agar that don't have calcofluor white, which don't grow back on the agar that do have calcofluor white, and you pick the former colony and analyze it further. So this very quickly got us to a nice collection of Zymoseptoria cell wall mutants. In total, we've got so far about 53 mutant strains based on hypersensitivity to 200 micrograms per mil calcofluor white. And interestingly enough, almost all of those strains also have a reduction in the switch to hyphal growth on yeast on water agar media. And interestingly, even more is that the majority of them also have a dramatic reduction in virulence on wheat leaves. So this is quite a nice way of linking potential cell wall defects to virulence defects. But obviously the question is, can you get to the underlying genes that are affected in the process? So what we did was sequence the whole genome resequencing again of 39 of those 53 available mutants. And this is a summary of the types of mutations and number of mutations we got in those strains. So when in total of the 39 strains we resequenced, we identified 186 genes across those mutants that had high effect loss of function mutations. So that averaged out just short of 5 genes were affected by a high effect loss of function mutation in each mutant. We obviously got more mutations causing moderate effects, which are a little bit difficult, certainly for our analysis. So a few thousand in total, and an average of 61 per strain. But, you know, that's not too bad. I was expecting a lot worse than that initially. So this is why we did it as a little bit of a look-see. It was still a question of whether we could make some sense of the genes that are affected by that, so those mutations, and whether we could nail it down to single genes being important for the calcifluor white hypersensitivity and the plant infection data. And overall, we have been able to. It's about a 50% success rate so far when we analyze the data, look at all the data we have in terms of expression data and other data sets as well for all the candidate genes. But we have been able to validate, and I just show you here, 7 genes in total that are specifically inactivated to cause cell wall weakness and loss of function. Loss of virulence, and I give you 3 examples there, different aspects of cell wall biology. Mannan polymerase generates the actual physical structure of the wall. KNR4 is likely a signal transduction integrator, and the Rho GEF protein is again a signal transduction protein. So this is looking good. We're starting to write this up as a pipeline, but the pipeline is very adaptable for screening mutants for anything. We've done it for calcifluor white, but you could do it for oxidative stress, fungicides, etc., etc. So it's quite a useful and powerful pipeline, I think. Which brings me nicely into the Abraham Wald essential gene screen and mutant collection, because it kind of builds on from the same data. So when we did that resequencing of the UV-induced mutations and looked at the numbers that we got, when you actually think about it, so if we sequence 39 individual mutants, we managed to get 186 genes that were clearly not essential for life, because the fungus would not have grown back and we wouldn't have been able to sequence it if they had been essential for life. So on typically, 4.95 genes could be inactivated by high effect mutation per strain using this approach, led us to the sort of idea to start to think whether it might be possible to generate sufficient UV mutant strains that if we resequence them all, would allow us to identify genes which could never be mutated. So never incurred a high effect mutation in the data set, indicating they could be putative essential for life genes. And this is very similar to the concept developed by Abraham Wald, which is the Abraham Wald survivorship bias screen, which he employed during World War II to study aircraft that returned successfully from missions, bomber aircraft. And he looked on the aircraft to see where they had artillery damage and also where they never had artillery damage. And the principle being that if a region of the aircraft never had artillery damage, it probably never made it home. So therefore it was of critical function for the aircraft. And basically that led to the Allies reinforcing certain areas of those particular aircraft to make them more resilient to potential artillery fire. So we thought whether we could do something similar for a fungal genome, just pepper it with random induced mutations and identify the genes that perhaps never ever could be mutated, so that we never found them mutated in viable strains, and therefore whether those genes are likely essential for the life of the fungus. So this is what we did. We actually UV mutagenized again our reference strain of IPO323, and we picked 25,000 colonies into 24 well plates by hand. It's a bit of a team effort. Grew them all up until they were saturated, and then we made pools of them for genomic DNA extraction. So we made 500 pools of 48 individual mutants, but we've also got a collection of each and every 25,000 mutant as a glycerol stock, taking half a -80 freezer and several half -80 freezers down in our basement. But we did, together with an industrial sponsor, we did sequencing of these pools. And basically the whole idea was to identify whether or not we could whether we could identify genes in the genome that had no or fewer than expected high effect mutations, and then to test and validate some of those predictions in the lab. So the top here is the global overview of the data that we got, which was kind of great. So this is just data for the high effect mutation rate relative to the predicted length of the protein or the gene, as it's expressed here. There should, in general, be, if there's no link between link between mutation rate and gene length, most of the data should fall on that line of expected mutations per gene length, and many does, as you can see. Those that are above the line are genes with higher number of mutation than expected, which for us, not of interest. But the ones that fell below the line are obviously of extreme interest, which are genes with lower numbers of mutations than you would expect, given the size of the predicted gene and protein length. So that was looking pretty good. And then we started to break this down into different categories of genes. You can see on average, the number of, for all proteins in the IPO323 genome, you can see the number of high effect indel mutations and high effect SNP mutations. Then we looked at the major facilitator transporters, which are known to be very redundant in function in Zymoseptoria. There's 400 of them, and those had a very high rate of indel and SNP mutations induced by the UV. But when we looked at the known and predicted essentials and current fungicide target genes, these genes came out with much lower levels of UV-induced mutations. So it sort of our original idea for what we tried to do seems to have worked. But we've gone on since then to do some experimental candidate validation of these candidate new essential genes. And what we use here is a technology we're able to do where we swap the gene of interest or with its own promoter. We effectively replace its own promoter with a promoter we can control under laboratory conditions. So we use the nitrate reductase promoter, which effectively facilitates growth on nitrogen. Growth on nitrate. The promoter is on, so switch the gene on. But if you grow the strain on more complex nitrogen media, it switches it off. So it's a growth no growth assay, if you like, for your gene of interest, whereby you effectively control it by replacing its native promoter with that promoter. The rest of the data just shows how we kind of select for strains. So we identify targeted promoter replacement strains by having only the single upper band with the green thing there in a PCR-based assay, whereas ectopic strains where our transgene has gone somewhere else and the wild-type gene is still there, you end up with the two PCR fragments, basically. And then when you identify those, you can start screening them for the on-off switch. And I'm giving you an example here of two genes that we have identified so far. I can't give you the exact details of what they are due to confidentiality, but one is a transcription factor and one is involved in the general process of protein ubiquitination. And you can see, starting with the left panel, when you take these mutants, so we have ectopic mutants, which are pink. They are controls. IPO323 is a control at the bottom. And then you have the greens, which are the validated promoter swaps, where we've effectively introduced the nitrate reductase promoter instead of the native promoter. And then when you switch those strains onto off media, which either contains an amino acid like glutamate or on rich media like YPD, hopefully you can see for both examples that those strains can no longer grow when those genes are switched off. So this is really good. It's kind of working reasonably well. We've validated 15 genes to date from the WALD data for being candidate essential for life genes in Zymoseptoria tritici, although there is some confidentiality on that at the moment due to the company being involved. What we're also able to do, of course, now going forward is that we have this fantastic resource of 25,000 mutants in our -80 freezer, and we've started doing high throughput screening of those, basically in 96 well plate assays for different stress responses to pre-screen them for then testing them on wheat, and again to resequence their genomes individually to identify candidate genes and validate them as novel targets and so on. And that's going to be a cornerstone of what we're going to do for quite a few years. Okay, the final part of the presentation is some slightly different work in about how we're using Zymoseptoria as a tool for discovery of new microbe-derived crop protection products. So Rothamsted research, we are quite diverse in the number of staff and colleagues we have, but we have many field trials spread over the areas of our different research stations, which are being sampled continuously, different crops, different environments, different treatment regimes, and they're being sampled continuously for many things, but also they're being sampled continuously also by our microbiologists who are establishing cultures of various different bacteria. But in this particular example, I show you examples of some of the collections they're making of pseudomonads. And these guys are known to have antifungal activities. This is through biocontrol and other types of assays in the past. So we thought we would just set up a simple assay, again, exploiting the yeast-like cells of Zymoseptoria tritici, so we could effectively form a cell lawn there, which you can see GFP tagged at the bottom just to be fancy. But you can make a cell lawn of Zymoseptoria cells, and then you can start spotting these little bacteria on the top of that lawn to see whether any create a zone of inhibition, which is a nice clear zone around where the bacterial spot has gone. And this worked really well in a PhD project. And we got 37 inhibitory bacteria through the assay, which we sent for whole genome sequencing. The idea was to start looking at secondary metabolism to see if they had the capability to produce any interesting secondary metabolites. And we compared those 37 again by sequencing 40 strains also that had no inhibitory activity in the antifungal assay as well as a control. We also have chemists at Rothamsted. We're very lucky, they're just down the corridor. And what they also did was generate LCMS traces from the agar surrounding the zone of interaction, if you like, the little halos that you can see there on the plate. So then we were entered the sphere of bacterial secondary metabolite predictions, and our student has generated a really nice pipeline using available resources that are out there, plus some new tools. And he was able, from his particular mini collection of 39 strains, to identify 120 putative gene clusters for secondary metabolites in the inhibitory bacteria, only 10 of which were known. So it meant that even from that quite small collection of strains, we have something in the region of 110 unknown secondary metabolites potentially being produced by those bacteria, some of which could be antifungal. We decided to tweak the assay as well. So as I started this presentation, I told you about how much genetic variation we can see between Zymoseptoria strains, and there's no point going down a fungicide discovery route if you at the end find your fungicide will only target 2 or 3 of those strains and not all of them. So we started screening all the bugs against different Zymoseptoria tritici isolates, which was very worthwhile because we could start to see that some isolates were more resistant to inhibition by some of the bacteria, whereas some were more sensitive. So then we started to focus down on those bacteria that were always able to give a good strong level of inhibition of all isolates of Zymoseptoria tritici, because this is yet another quantitative trait in the fungus, basically, this sensitivity. And as a proof of principle, what we did identify in the genome of some of these inhibitory bacteria were a biosynthetic gene cluster that is likely to produce the known product DAPG, so 2,4-diacetyl fluoroglucanol, which has been shown to be antifungal in various different assays and historically against various different species. We were able to identify that particular cluster reproducibly in strains that inhibited all Zymoseptoria isolates. We were able to find it in the genome, and we were also able to identify the peak in the LCMS for 2,4-DAPG in the zone of inhibition. But we still had to prove or test as a proof of concept that it was DAPG in those bacteria that were responsible for the antifungal activity. So we've developed mutagenesis in bacteria as well now, which is quite nice. And this is the example of what we did. So we targeted by mutagenesis inactivation a key DAPG biosynthetic gene called PHLD. And when you knock that particular gene out in 2 of the inhibitory bacterial strains, you can see on the slide there, On the right-hand side, you can see that they lose their halo. They lose their ability to inhibit the fungus. So quite clearly, DAPG was the secondary metabolite produced by that, which was giving such consistent control of the fungus in an in vitro assay. And our pipeline now is obviously focusing more on some of these unknown, these new products that we've got from the genomic bacterial predictions. But an obvious question for about DAPG, which is something that I found curious, was that so what is it? Can it be used as a crop protection product in itself, or can the bacteria that make it be used as a crop protection product? So we did a kind of biocontrol and a chemical control assay. In summary, the biocontrol assay, so actually putting the bacteria on the wheat leaf with the fungus, these are bacteria that produce the DAPG, were unable to control the fungus. So they were obviously not resilient to the leaf environment, not producing DAPG or whatever. We don't quite know why yet, but it doesn't work. Whereas the chemical control using DAPG itself was giving us a dose response relationship for protecting leaves against Zymo. So overall, I think this supports our general approach that we have for this project, which is to go using the genome mining of the bacteria to go after the secondary metabolite clusters and go after the chemistries themselves, rather than using the bugs as biocontrol agents. I think that might be more difficult, but perhaps not impossible. It might be more difficult. Okay, I think that was my last slide. I'm just going to summarize there and give some perspectives. I hope what I've shown you is that the genetic diversity that you can see within Zymoseptoria tritici populations can actually be used against it. If you deploy pangenomics that focuses down on what is core and what cannot be mutated at a high rate with it by individuals in the population. Hopefully, I've also shown you, which we've known for some time, that the outcome of the fungal plant battle, if you like, is almost dictated by the fight between two fungal chitin binding proteins. And the fungal protein is currently winning that against the plant receptors. Also, what I hope I've shown is that the yeast-like morphological growth phase is a super tool for many of the work we do on Zymoseptoria tritici for generating mutations, populations, etc. And it's quite unusual for a haploid filamentous plant pathogen, so it makes it a really good system. Fungal cell walls are a valid target for future methods of intervention, I believe. And in terms of going forward, I mentioned right at the start my thing about all these genes of unknown function and how can you actually prioritize them and motivate yourself to work on those genes. Well, what we're currently doing is combining these two large data sets of the protein-specific natural variation we can see, pangenome data, with our induced Abraham Wald data to look at each individual encoded protein of unknown function and to see what is mutating on a very low rate, potentially in both data sets. And we're going to prioritize those for some functional analysis to see if we can start revealing some of the functions of the dark matter of fungal genomes. Okay, that's it. I'd just like to acknowledge everybody whose data I presented specifically today. That's on the left is my Rothamsted team from now and before, PhD students, technicians, and bioinformaticians. We've been lucky enough on two of the major projects to have support from Syngenta and Bayer. And obviously, Bart Thoma was a key collaborator for us in all the work we've done on the lysine effectors. And the image there that I finished with shows the GFP tagged Zymoseptoria tritici emerging from stomata of a destroyed wheat leaf and getting ready to infect other green areas of the plant tissue. Okay, thank you very much. I'd be happy to take any questions if there are any.
Thanks, Jason. Yeah, if anyone has any questions, please feel free to put them in the chat or unmute and ask. If not, Jason, I have a question about the work that you presented in the beginning on the chitin binding protein, gosh, what was it? CT3 lysem.
Yeah.
For the transcriptomics, your sampling began 6 days post inoculation.
Yeah.
And I was curious, what role do you think that protein plays in the latent phase? And if it does, how early would you expect to see differences in expression?
So I did, just for simplicity, I didn't show. So basically, Luca, the PhD student that took on this work, well, she made some 3-lysm promoted GFP expressing strains. So we actually started to look at when the fungus lights up when you start inoculating wheat leaves with it, and it's on super early, within a few hours actually. So I think the expression of that particular gene may be linked in with the transition to hyphal growth. Basically, I think it might be baked in. The fungus knows it's going hyphal, and it may just be switching that on at the same time. I don't know whether they're separable.
Okay.
Very early.
Yeah, interesting. Gillian, you had raised your hand.
Yes. Can you, nice talk.
Thank you.
Can you comment in general about genes and gene structure on dispensable chromosomes?
Yeah, so basically, there are some key features. A lot of the predicted genes on the dispensable chromosomes, a lot of them have a lot of evidence for RIP. So they look like they're inactivated, duplicated copies of things that are sometimes on the core chromosomes. I think there's also more epigenetic silencing on them as well, which is probably responsible for the very low level of expression of them. And very few of them have obvious predicted functions. They tend to be smaller than the genes on the core chromosomes, and there's very few really clear predicted functions on them. So obviously many of our colleagues are very interested in this. I'm interested in this as well, but my goal is to identify the things that are really important for virulence. So I tend not to spend too much time looking at them apart from superficially.
Thanks.
Jason, I had, I suppose, not a question, more so a comment that I'm feeling inspired by your results in the last component, revealing that when applying bacterial cultures directly to leaves, you're in many cases not getting effective management of a disease. And so I think for a lot of us who are trying to identify biocontrol products to manage diseases, we're often using products that say they have active cultures, and you're waiting for colonization to occur on the foliage in order for bacteria to produce the secondary metabolites and other products. And so perhaps if we look instead for products that just have the chemical of interest and bypass the need.
Yeah, that's our angle basically. Just because that's the way we are set up. We've got chemists, we've got microbiologists, bioinformaticians. We just think that this is something that we can run with in terms of natural product discovery pipelines rather than biologicals, basically. I mean, the data I've shown you is obviously just a couple of different bacteria. That doesn't mean to say it won't ever work or anything like that. That's just what we've tested so far. But it does reinforce what we thought would be our better approach, or our preferred approach was to go after the chemistry if we can, basically, and how the microbes make this stuff and what they are, and to see whether they can be synthesized or in another way to be tested as a natural crop protection product, basically. Yeah, I think it's a bit. I think the wheat leaf obviously is a pretty barren place for a microbe to live. Maybe, I mean, we have started now generating collections of bacteria directly from wheat leaves, so they should be able to persist there. At least that is their natural growth habitat. Whether they will fare any better or whether they will have fungal inhibition capability, we'll find out in due course. I think there was something in the chat. I don't know whether something popped up.
Oh, yes. Yeah, so we have a seminar room in a building, and they're saying, I particularly appreciated thinking about the essential genes through the lens of the Abraham Wald metaphor.
Yeah, thank you. Yeah, it's fun. It's been fun, that one. Good project.
Oh, and yeah, there's a question in the chat. Do you think the fast forward genetics pipeline could work for other closely related species, or is it specific for this pathogen?
Basically, the way we've set it up is we're taking advantage of this sort of yeast-like growth we can have on rich nutrient agar. That's what allows us to print onto velvet and to effectively treat it like a yeast. I think if you have a very filamentous pathogen, it would be more difficult to get the throughput that we can with a more yeast-like pathogen like Zymoseptoria. So if you can manipulate your pathogen to not be too highful under a particular growth condition so that you can print it and make replica plate it easily enough at scale, then it should be possible.
Well, yeah, if there are no other questions, I'd like to thank Jason again for taking the
you.
time and presenting during your dinner hour. And
It's coming up now. I'm feeling hungry.
Yeah. Yeah, thank you for the seminar.
No problem. Thank you very much for the invitation, and have a nice afternoon, everybody.
Thank you.
Bye-bye.
This has been a production of Cornell University, on the web at cornell.edu.